Hello my dear friends, welcome to live prep. So in this lecture we will talk about overview of the cell. So in the NCERT they have given the heading of overview of the cell, but actually there they have discussed only about eukaryotic cell. They have not discussed about prokaryotic cell. But here in this lecture I will discuss about both prokaryotic cell and eukaryotic cell. Separately I will discuss, so that from the next lecture onwards, anyhow we are talking about the prokaryotic cell. So at that time it will become very easy to understand the NCERT content. Okay. So quickly we will go with the overview, because anyhow individually for each component separately we will study the content. So simply it is an overview. So what are the various components that are present? If we understand that is enough. So in this way, first we will get aware of what are the different names that we are using for the different cells. All the things we will understand first. And first we are talking about prokaryotic cell, and then we will discuss about the eukaryotic cell, and then finally we will read the NCERT content whatever they have given, so that we will conclude this particular lecture, so that from the next lecture onwards we can go with prokaryotic cells. Okay. First I will show the first I will give the explanation for the complete content, and then quickly we will go through this PowerPoint slides. Okay. So first we are going with prokaryotic cell, overview of the prokaryotic cell. So whenever we are talking about a prokaryotic cell, so generally we need to get one particular doubt. And what is that particular doubt? And this is very, very important. So please remember this. Under the prokaryotic cells, we are studying about different types of bacteria, mycoplasma, many are there. But here I am focusing about two things. One is regarding eubacteria. One is regarding eubacteria, and other one is regarding archaebacteria. Eubacteria and archaebacteria. Anyhow, you will study separately about these things under the biological classification chapter. So there you will know that there are huge differences between eubacteria and archaebacteria. That is the reason why in the 3-domain classification, eubacteria was kept separately and archaebacteria was kept separately, and all the eukaryotes were kept separately. So in this way, 3-domain classification is clearly demarcating between eubacteria and archaebacteria. But I already told you that NCERT is following 5-kingdom classification. In the case of 5-kingdom classification, we are combinedly studying this under kingdom Munira. Yes or no? So that you will get some doubts for different sentences. So I am making it very clear in advance. What is that particular clarification? Whenever we are talking about prokaryotes, in general, we are talking about eubacteria only, because for majority of the characters, archaebacteria is differing with eubacteria. So in that case, you may get the doubt. So in general, prokaryotes means we are talking about eubacteria only. Understood or not? If you want to say something about archaebacteria, in that case, we will clearly mention that particular point, or else NCERT will clearly say that in archaebacteria, so and so. In that case, no problem. In general, they are talking about prokaryote means they are talking with respect to eubacteria. This point you need to keep in mind. Please don't make any mistake here, because in class 11th, class 12th, many things related to the prokaryotes we will study. Wherever we are talking about prokaryote, it is in general sense, it is with respect to eubacteria. This point you need to compulsorily remember. Okay. So anyhow, whenever we are encountering these particular things, once again I will tell, but still you need to keep this in mind. And now we are talking about prokaryotic cell. What is the meaning of this pro? Pro means what is it? So in the case of software and all, we will say that pro means what? Professional there. In that case, professional, higher level. But in this case, pro is not higher level. It is the previous form, before form. Before making something, whatever is the previous form is there, that is called pro. And then carrion. Carrion. What is this carrion? Carrion means nucleus. So it is a prokaryotic cell. That means it is a cell that is not even having complete nucleus. Please understand this. It is not even having complete nucleus before forming the nucleus. So there is some other form that is called a nucleoid. So nucleoid containing cell. That's why it is a prokaryotic cell. But what about eukaryotic? Eu means true. True carrion means nucleus. True nucleus containing cell. That's why those cells are called as eukaryotic cells. But in these cells, complete nucleus is not present. And the same meaning you can derive in a different way also. So what is this particular derivation? So you need to focus on evolution of the organisms. Anyhow, in the evolution separately we will study in class 12 chapters. So in the evolution, what happened? Initially formed cells were prokaryotic cells. So first prokaryotic cells were formed. So after, in the course of evolution, slowly from the prokaryotes, eukaryotes were developed. So in the eukaryotes, well-defined nucleus was developed, and here well-defined nucleus is not present. So it is the before form for that particular nucleus. So that we are observing in the prokaryotic cell. So that's why in this way we are calling this as a prokaryon, before form for the nucleus. And here well-defined nucleus is not present, nuclear membrane is not present, chromosomes are not present, directly nucleoid is present. And where this nucleoid is there? Directly in the cytoplasm. Anyhow, some of the points we will discuss in overview, and the clear-cut details we will study separately in prokaryotic chapter. Okay. So in the prokaryotic part separately we will study about that. So in this way, it is the before form for what? Nucleus. And that type of content is present in this cell. That's why it is called as prokaryotic cell. In general, we are studying for eubacteria. So with respect to that, we will study about this particular part. Is it clear? And now we are starting with overview. So now first we will see what are the various components that are present in prokaryotic cell. This is the contents. What are the various contents that are present? If we see that, overview is completed. And what is the role of each and every component? That separately we will study. Okay. So in this overview of the prokaryotic cell, what we need to study here? Observe this carefully. For any cell, whether prokaryote, eukaryote, whatever it is, if it is a cell, means compulsorily it should consist of cell membrane. Cell membrane. What is the other name for cell membrane? Cell membrane is also called as plasma membrane. So it is compulsory, whether prokaryotic cell, eukaryotic cell, whatever it is. If we are saying that it is a cell means compulsorily plasma membrane should be present. Cell wall may or may not be present, but cell membrane or plasma membrane should be compulsorily present. So that's why first we are talking about cell membrane. And now in the case of prokaryotes, what is the sentence they are saying? Most of the prokaryotes are having cell wall, but only few types of prokaryotes are not having cell wall. What are those that are not having cell wall? Already we have discussed in the previous lecture. We studied that they are the smallest cells. Among all the cells, all prokaryote, eukaryote combined, you just see smallest cells are mycoplasmas. They are also prokaryotes. But for the mycoplasmas, they do not have cell wall. Already we studied that in the previous lecture, right? They do not have cell wall. So for mycoplasmas, what is the outermost layer? Plasma membrane is the outermost layer. But here we are talking in general for the eubacteria. In the eubacteria, what happens? There is a cell wall that is present around the cell membrane, and this layer is called as cell wall. It is called as cell wall. And now you need to notice a small important thing here. What is that? Notice this is the cell membrane, this is the cell wall. And when I have drawn this particular cell wall, did I attach plasma membrane and cell wall each other, or did I leave some gap here? This point also is very important. So deliberately I left some space here. So that means between cell membrane and cell wall there is a space, there is a gap, here, and this gap is present around the plasma membrane. So around means it is peri. Please understand these words. Peri means what? Around what? Plasma membrane. So periplasmic space. So in this way, the space or the gap that is present between cell membrane and cell wall is called as periplasmic space. Understood or not? This is what about the periplasmic space. So that's why whenever we are drawing these two, we are not attaching both of them. We are leaving some gap here, and this gap is called as periplasmic space. Very good. And then outside the cell wall, there is one more layer, and that layer is called as glycocalyx. But how we are drawing this particular glycocalyx layer? So will you keep some gap and then will you draw glycocalyx here? No. And here how we are drawing this? Attached to cell wall, attached to cell wall. So to say that attached to cell wall, I am drawing the layer like this. So that means there is no gap here. So that means glycocalyx is completely attached to cell wall. That means there is no space between glycocalyx and cell wall. And let us write this one as glycocalyx layer. Glycocalyx layer. So each layer function separately we will study under prokaryote. So simply here you just understand what are the various names, that's it. So in this way total it is having 3 layers. What are the 3 layers here? Cell membrane or plasma membrane. Outside to this cell wall is present and outside this glycocalyx is present. So total 3 layers. And these 3 layers combinedly it is called as cell envelope. So prokaryotic cell it is having cell envelope. It is having cell envelope. So in this cell envelope what are present? Total 3 components are present. 3 components are present. And these 3 components we will write from outer to inner. So sequence from outside to inside we will go. So what is the outermost? Glycocalyx. So outermost is glycocalyx. And this glycocalyx is followed by, already I told you the use of these words, followed by, follows, preceded by, precedes, all these things, yes or no, in the general introductory lectures. So there I told about all these things, how to use these English words, and in the NCERT words they have given some sentences, how to understand this. So there in that lecture I told you how to understand these particular words. So here followed by means next one. So glycocalyx, from outer to inner, glycocalyx is followed by means next one. What is the next one here? So glycocalyx is followed by cell wall. So we need to write cell wall here. And now the next one, this cell wall is followed by, what is the next layer here? So the cell wall is followed by cell membrane or plasma membrane. So it is followed by cell membrane or plasma membrane. So in this way, these 3 combinedly it is called as cell envelope. So this is part of prokaryotic cell. And not only this, and this cell membrane or plasma membrane is enclosing some content. And what this content is called as? In the case of eukaryotic cell, we are using a term called protoplasm. But in the case of prokaryote, directly we are using the term called as cytoplasm. So it is also having a semi-fluid matrix, it is called as cytoplasm. Semi-fluid matrix, that is called as cytoplasm. Please understand this. What this semi-fluid means? Semi-fluid, not completely fluid like, not completely solid like, it is in between this. So that's why it is called a semi-fluid matrix that is surrounded by which one? Cell membrane or plasma membrane, that is called as cytoplasm. But in the case of in the case of eukaryotes, we are using directly protoplasm term. So there nucleus is present. So that's why there we will study the separate thing. But here cytoplasm term we are using. And in this cytoplasm, is there any nucleus in the case of prokaryotes? Nucleus is not present. Then what are the various components that are present in this particular cytoplasm? These things we need to write very clearly. Okay. So what this cytoplasm is consisting of? In this cytoplasm, many things are present. For our understanding purpose, I am writing each one separately. Anyhow, when we are studying about prokaryote, regarding each one separately, we will study. Okay, in the same way, we are studying all the things. Okay, so what are present in this? Definitely, some fluid matrix is present. Only fluid part. Okay, so only fluid part. If you are talking, it is having the fluid matrix. And what is this fluid part is called as? This fluid part you can call it as cytosol. Only fluid part you are calling this as a cytosol. Okay, so it is cytosol. And not only fluid part, it is also having some other things. What are the other things? It is also having DNA. It is also having DNA. Where this DNA is present? It is present directly in cytoplasm. Yes or no? So this is present directly in cytoplasm. And what is the name for this DNA? And here this DNA is again of 2 types. What are these 2 types? One is compulsory. One type of DNA is compulsory for every bacterial cell. If this is not there, cell will die. So that's why this is compulsory. What is compulsory? Nucleoid is compulsory. Nucleoid is compulsory. Okay. So what is this compulsory part under this DNA? One is nucleoid. And not only this, some bacteria, please remember I am using the word called as some bacteria or certain bacteria. This wording is very important. Anyhow, regarding this, when we are studying about prokaryotic cell, I will clearly explain all these things. So please understand this. Compulsory is nucleoid. Nucleoid is compulsory for each and every prokaryotic cell. But along with this, some bacteria or certain bacteria. It is not all bacteria, not all bacteria. It is not compulsory for all. Some or certain bacteria are having some extra double stranded DNA. Okay. Anyhow, properties, everything separately we will study. Some extra DNA also present. And this extra DNA is called as plasmid. This extra DNA is called as plasmid. Very, very important content. So please remember this. So in this way, this DNA, compulsory part is nucleoid and optional is plasmid. And some bacteria are having another. It is not that if nucleoid is not there, only plasmid is there. No, they cannot survive. Nucleoid is compulsory. Whether plasmid is present or absent, it is a different case. But nucleoid is compulsory. So nucleoid is compulsory, and here in some bacteria, plasmids are also present. This is what the point that is related to DNA. So in this way, within the cytoplasm, DNA is also present. Not only DNA, it is also having some other things called organelles. What are these organelles that are present in cytoplasm? Organelles. What type of organelles are present? Are they membrane bound organelles? No. In the case of eukaryotes, membrane bound organelles are there. But in the case of prokaryotes, membrane bound organelles are not present. And here, in the case of prokaryotes, only one type of organelles are present. Only one type. And what are these type of organelles? They are called as ribosomes. And these ribosomes are present in eukaryotes also. So that is the reason why ribosomes are the organelles. Ribosomes are the organelles that are present in all the cells. All the cells means both prokaryotes and eukaryotes. All the cells are having ribosomes, and that too ribosomes are non-membrane bound organelles. That means they are not surrounded with any membrane. And what is this concept of membrane? Slowly we will study. Anyhow, here it is not needed. So in the next lectures we will study about that. So within the cytoplasm there are some organelles called as ribosomes. So these are also present in the cytoplasm. So cytoplasm is also having some organelles. And what are these organelles? Only type, they are called as ribosomes. Only ribosomes. No other organelle. In the case of eukaryotes, endoplasmic reticulum, Golgi complex, lysosomes, vacuoles, mitochondria, plastids, likewise many are there. And where they are present? In eukaryotes. But in prokaryotes, only ribosomes. And these ribosomes are present even in eukaryotes also. So that's why ribosomes are the non-membrane bound structures present in all the cells. All the cells means both prokaryotes, eukaryotes. The same sentence you can also write in a different way. How you will write? Present in all the 5 kingdoms. Yes, you can say it's all 5 kingdoms. Or you can say that present in both unicellular and multicellular organisms. Anyway you can say it is correct. And ribosomes are present in all these living organisms. Then what about viruses? Do viruses have ribosomes? No, they don't have. We are not considering them as cells also, right? So that's why this is about ribosomes. So next one is not only ribosomes, they are having some other granule like some sand is, if you are touching a sandule, it will be appearing like a small granule, right? So in this way some granular structures are also present, and these granular structures that are present in cytoplasm are called as inclusion bodies. And these are of different types. Anyhow, when we are talking about prokaryotes, there we will study. So inclusion bodies are also present in this cytoplasm. So here we need to write one more, that is inclusion bodies. So in this way, these are the major ones that we are talking about a prokaryotic cell. In the prokaryotic cell, now let us quickly go through the overview. What are the various layers present? Outermost glycocalyx, that is followed by cell wall, and there is no space between these two. And then there is a space here, and that space is called periplasmic space. After the space, inner to the cell wall, then what is present? Plasma membrane is present. And in this way, plasma membrane, cell wall, glycocalyx, all the three combinedly it is called as a cell envelope. Overall function of cell envelope is protection. Not only this, individually they are having specific functions that we will see separately. Clear? Now, and this cell membrane or plasma membrane is enclosing some semi-fluid matrix, and this semi-fluid matrix is called as cytoplasm. And this cytoplasm is having some only fluid part. Only fluid part is called as cytosol. Not only cytosol, it is also having, cytoplasm is also having DNA. This DNA may be in the form of nucleoid or it may be in the form of plasmid. But here what is compulsory? Nucleoid is compulsory, but plasmid is optional. Some bacteria are having plasmid, but some are not having plasmid. But all the bacteria are having nucleoid. This is compulsory. And along with this, they are also having some organelles like ribosomes. These are non-membrane bound organelles present in both prokaryotes and eukaryotes. And not only this, they are also having some inclusion bodies. Generally, these are some food storage or different other molecules that are stored in this particular one. And not only this, in the case of some photosynthetic aquatic forms, even gas vacuoles are also there. But anyhow, don't worry about these names. Simply remember some inclusion bodies are there. So this is the overview of the prokaryotic cell. Understood or not? With this, overview of prokaryotic cell is completed. Now we are going with the next one, that is overview of eukaryotic cell. Overview of eukaryotic cell. In the case of eukaryotic cell, what is this? U means true. U means true. So true carrion means true nucleus is present. So the cells that are having the true nucleus, what is true nucleus? Membrane bound nucleus. Membrane should be present. Nuclear membrane should be present. Inside this chromosome should be there. Inside the chromosomes DNA should be there. In this way, if it is present, it is called a true nucleus. So since these cells contain true nucleus, these cells are called as eukaryotic cells. Anyhow, more description we will study separately under eukaryotic cells. This is just an overview. So that's why I am giving the word and its meaning. So this is the overview. Now in this overview, what we need to study here? So here we need to talk about eukaryotic cell. Eukaryotic cell and its component. Eukaryotic cell and its components. In the eukaryotic cell, now here also we will make a diagram first, and then from that we will write each and every component. So in the case of eukaryotic cell, now here there is a small problem. So generally here for our understanding part, so on one side we will talk about plant cell, whereas on the other side we will talk about animal cell. Plant cell and animal cell. Now what is the difference here? In the case of animal cell, since it is a cell, compulsorily plasma membrane is present. This is the cell membrane or plasma membrane. And here also in the case of plant cell, there is a plasma membrane. For understanding, I am drawing this particular rough diagram. So plasma membrane or cell membrane, you can also call this as cell membrane. Understood or not? Yes, in both the case it is present, but where you are getting the difference? With respect to the cell wall. Please understand this. Here, in the case of plant cell, there is a cell wall that is present on outer side of this plasma membrane. Please understand this. Then what about the cell wall here? In the case of animal cell, cell wall is not present. So in this way, now we are getting some differences here. So in the case of eukaryotic cell, generally we are not talking about glycocalyx. Please understand this. In the NCRT, nowhere they are talking about glycocalyx for eukaryotic cell. They are talking about glycocalyx for prokaryotic cell only. But in higher classes, you will study that attached to plasma membrane, some carbohydrates layer is present. And in the case of animal cell, that layer itself, not all cells, majority of the cells are having. So if carbohydrate layer is present attached to it, that itself is called as glycocalyx. Glyco means what? Carbohydrate. It is a layer that is mainly having carbohydrates, that is called glycocalyx. Anyhow, slowly we will understand all these things. So in some of the animal cells they are having, but at the level of this particular one, do we need to mention this or not? No, we are not mentioning. We are not talking about glycocalyx anywhere in eukaryotic cell. So simply ignore that part. Maybe in higher classes you will study about this. Clear? So in the case of animal cell, the outermost layer will be what? Cell membrane. Whereas in the case of plant cell, what will be the outermost boundary? In this case, it is a cell wall. Cell wall is present. That's it. And in the case of eukaryotic cell, only these will form the cell envelope. So now we are talking about eukaryotic cell. For the eukaryotic cell, when we are writing this, here also we are writing cell envelope. Here also we are writing cell envelope. But in this cell envelope, we are writing only two components because we are removing this glycocalyx. We are not talking about glycocalyx. Directly we will talk about cell wall. Cell wall. But is this cell wall present in all the organisms? No, it is not present in all the organisms. Cell wall we are observing in fungi. Fungi will have the cell wall. and also planty. Planty means what? Plants. Plants are having the cell wall. Then what about the protista? Do all the protists have cell wall? No. Some protists are having cell wall. Some protists are having cell wall. That's it. That means remaining protists are not having cell wall. And what about the kingdom Animalia? Animals are not having cell wall. So, if you are talking about this, then only you will talk about the cell wall. If you are talking about the animals, we will not talk about the cell wall. So, here next one will be cell membrane, and this is compulsory for all. So, whatever is the organism, whether prokaryote, eukaryote, whatever it is, cell membrane is compulsory. So, in this way, in the case of eukaryotic cell, so cell wall and cell membrane. Cell membrane is compulsory. In some of the cells, cell wall may be present. So, this is what about the cell envelope. So, cell envelope that is related to this eukaryotic cell. So, this is what over. And now we need to talk about the content present inside this. And from here onwards, for both of them, we are talking about the same thing. Is it clear? So, now directly I will show in this diagram only, so that we will go for this. Now, this cell membrane is enclosing something. Now, all the contents that are present inside the plasma membrane, now here it is called as protoplasm, not cytoplasm. Here we are changing the word. What is the word that we are using here? It is a protoplasm. It is a protoplasm. Protoplasm. Now, what is the difference between protoplasm and cytoplasm now? Now, you need to focus on these particular content. In the case of eukaryote, directly I am talking here only. I am not talking about that because inside content is almost similar here also. Now, here in the case of eukaryote, there is a nucleus. There is a nucleus. And now what we are saying, generally when we are talking about cytoplasm, how you will define cytoplasm? Cytoplasm means between nuclear membrane and plasma membrane, whatever is present between nuclear membrane and cell membrane, and this is called as cytoplasm. Cytoplasm. Understood or not? But what is protoplasm? Entire cytoplasm plus nucleus. Entire cytoplasm plus nucleus. That means everything that is present inside plasma membrane will be protoplasm. Completely it is a protoplasm. From this protoplasm, if you are eliminating the nucleus, only the nucleus if you are separating, whatever is remaining, that part is called as cytoplasm. So, how you can write protoplasm? We can write protoplasm as protoplasm is equal to, how we can write this? Protoplasm is equal to cytoplasm plus nucleus. And sometimes what they will say, content present inside the nucleus is called nucleoplasm. So sometimes it is written as cytoplasm plus nucleoplasm is equal to protoplasm. Now based on this you can write, then what is cytoplasm? Protoplasm minus nucleus. That means from this entire protoplasm, if you are eliminating the nucleus, whatever is remaining, that is called as cytoplasm. Then what is this nucleus or nucleoplasm? Protoplasm minus cytoplasm, whatever is present, that is called as nucleus or nucleoplasm. So in this way also they are giving one particular definition. So please understand each and every point. So they, like this also they may ask in examination. Anything is possible in examination. So you need to understand the concept carefully. So this is what about the protoplasm. And before going into internal details, I want to make you aware about one particular word, and that word is protoplast. That word is a protoplast. Please understand the word. It is not a protoplasm. It is a protoplast. What is a protoplast? Now tell me here. What is a protoplast? What is the difference between a protoplasm and this protoplast? Now understand carefully. Protoplasm means this inside content. Now this inside content, that is called protoplasm, and this protoplasm plus only plasma membrane. Don't include the cell wall, only plasma membrane. Protoplasm plus plasma membrane. And these two combinedly it is called as a protoplast. Please understand this. So, in the case of animal cell, whatever the cell we are saying, that itself is called as protoplast, because protoplasm plus plasma membrane. So, animal cell itself is equal to protoplast. And the same logic apply here. And now they may ask a question in a different way. What is the question? Listen the question carefully. Suppose I am an examiner, I am giving you a plant cell. I gave you a plant cell and I am asking you to prepare a protoplast for the plant cell. Then how you will prepare a protoplast for the plant cell? Very simple logical question. So I gave you a plant cell and you need to prepare a protoplast for the plant cell. Then how you will prepare this? What is protoplast? Protoplasm plus plasma membrane. This is called a protoplast. But in the plant cell, what is extra present? That extra is cell wall. So how you will prepare the protoplast? By removing the cell wall. So how you will remove the cell wall? Anyhow, slowly we will study and understand all these things. Directly I will give you the answer. Plant cell wall is composed of cellulose. So if you are treating this with cellulase enzyme, this cellulosic cell wall will be removed so that plant protoplast will be prepared. So now what is the question here? How you are able to prepare the plant protoplast? What will be the answer? Treat the plant cell with cellulase enzyme so that cell wall will be removed. So in this way, plant protoplast will be prepared. So in this way, each and every point is a logical and a technical. So don't try to by heart this, try to understand this. So in this way, the term protoplast is used. So what is protoplast? Protoplasm plus plasma membrane. In the case of animal cell, cell itself is called as protoplast. But in the case of plant cell, cell wall should be removed to prepare the protoplast. I hope you understood this. So this is what about the protoplast concept, and now we are talking about the protoplasm. So what is this protoplasm? So it is the complete content that is present inside or surrounded by plasma membrane. That is called as protoplasm. Now in this protoplasm, first we will focus on the nucleus. So anyhow, we need to write this. Now this protoplasm we need to divide into two parts. One is cytoplasm. And the other one is nucleus. So these two combinedly it is called as protoplasm. Now first we will talk about the nucleus. What were the sentences given in NCERT? This nucleus is a membrane bound structure. It is a dense structure. So that you can clearly observe this in a cell. So it is a dense membrane bound structure. How many membranes are there? Two membranes are present. And this information separately I am writing here. So what are present in the nucleus? Now let us make a rough diagram here. Suppose this is the cell. And now we are focusing about the nucleus. Regarding the nucleus, what is the most important thing? It is having nuclear envelope. Nucleus has nuclear envelope. In this nuclear envelope means what? Cover. It is the envelope, cover for the nucleus. And this nuclear envelope has how many layers? In this particular one, it is having two membranes. It is actually having, even though we are drawing like this, actually it is having two membranes. It is a double membrane bound. Double membrane bound organelle. So nucleus is a double membrane bound. That means it is having two membranes. What are these two membranes? One is the outer membrane of nucleus. Other one is inner nuclear membrane. Outer nuclear membrane and inner nuclear membrane. In this way, two membranes are present in the nucleus. And this is combinedly called as nuclear envelope. This is one important thing that you need to remember. And next one, what else is present inside this nucleus? Inside the nucleus, there is a semi-fluid matrix is present. Semi-fluid matrix or semi-solid matrix present inside the nucleus is called as nucleoplasm. This semi-fluid part is called as nucleoplasm. Already we wrote here, right? So inside the nucleus, the fluid part is called as nucleoplasm. So in this way, nucleoplasm is present. Not only this, there are some non-membrane bound. There is no membrane, but clearly you can see some spherical shaped structures inside the nucleus. And these spherical shaped structures present inside the nucleus are called as nucleolus. For the cell, it is the nucleus. For the nucleus, it is nucleolus. So in this way, it is present. Function separately we will study. Okay, it is the nucleolus. And not only this, In the case of cell cycle, we will study about interphase and division phase. In the case of interphase, there are some long thread-like structures. Some of them are passing through nucleolus also. Okay, so there are some long thread-like structures that are present, and these long thread-like structures are called as simply chromatin. These are called as chromatin. Chroma means color, tini means threads. They are colored threads. Why they are getting color? Do they naturally have the color? They don't naturally have the color. We are adding some colored molecules called stains or dyes. Slowly we will talk about this, so that they are getting color, and they are long thread like, that's why they are called as chromatin. In the interphase, they are present as thread like chromatin, but during the M phase, the same chromatin. So now in the interphase, long thread like, now it will undergo condensation, it will decrease in length, and they will form short rod like structures called as chromosomes. The same chromatin is also called as chromosome. That means chromosomes are present inside the nucleus. Very important. And now you just recollect, when we studied about prokaryotic, just now we studied no prokaryotic. Did I use the term chromosome there? No. Please understand this. The term chromosome is very important. It is used only for eukaryotic cell. We should not use for prokaryotes. That's why chromosomes are present only in eukaryotes. That too, they are always present inside this nucleus. Understood or not? Now in this particular chromosome, what is present? DNA is present. So DNA is the genetic material. In the prokaryote also DNA is the genetic material, eukaryote also DNA is the genetic material. But in the case of prokaryote, DNA is not present as chromosome. But in the case of eukaryotes, DNA is present as chromosomes. This is the most important point you need to remember. So this is the overview for the nucleus. So nucleus is having double membrane bound envelope is present, and inside this it is having semi-fluid matrix that is called nucleoplasm, and also nucleus is having some spherical structures called as nucleolus. Singular is nucleolus. If there are many, plural is called as nucleoli. Slowly we will study all this. And along with this, in the interphase it is having long thread-like structures called chromatin, and during the M phase of cell division, they are undergoing condensation, and then they are called as chromosomes. So in this way they are present, and inside the chromosome, genetic material is present. What is the genetic material? DNA is present. On the DNA, genes are present, and these genes will control all the cellular activities. All the activities of cell are controlled by the genes. Anyhow, slowly we will study about all these contents in the next coming lectures. So this is the overview of the nucleus. So in this way, first we have completed about the nucleus. Okay, this is about the nucleus, and this is a double membrane bound organelle. Double membrane bound organelle. Double membrane bound organelle. Double membrane bound organelle. This point deliberately I am writing because here also we will get some double membrane bound, and there for comparison it will be useful. Okay, anyhow, remaining content we have completed. Now come to the cytoplasm. Now inside this cytoplasm, what are present? So many things are present in this. Here also there is a fluid part. That fluid part you can call it as cytosol. Cytosol. So here also this cytoplasm is having cytosol. Cytosol is present. Yes or no? In the case of prokaryote, directly in the cytoplasm DNA is present, but here it is separately present in nucleus. Well defined nucleus is present. So that's why these are eukaryotic cells. And then we are talking about the organelles. Cytoplasm is having some organelles. Different types of organelles are present. This information is important. Regarding the organelles, separately I will write here, okay, in a separate slide. What are the different organelles that are present here? So that organelles part separately I am writing here. So please observe carefully. Organelles. What are these organelles? How we are classifying these organelles? So this is a rough diagram for the diagram purpose, and this is the nucleus, double membrane bound. Okay. Generally whenever we are writing nucleus, we are not writing two layers, single layer only we are writing, but anyhow you can understand this. Okay. But for clarity, here I have written two layers. Okay. It is having two membranes. Nice. What are these organelles and how we are classifying these organelles? We are classifying these organelles into different types. What are these? First one is double membrane bound organelles. Double membrane bound organelles. double membrane bound organelles. Like the nucleus, these are also having two layers. These are also having two membranes. Two membranes they are having. What are these double membrane bound? So now observe this carefully. First one is regarding mitochondria. Mitochondria, this is the inner membrane. This is the outer membrane. Okay. So in this way, what is the list here? First one is mitochondria. Mitochondria will come under this. Okay. And not only this, in the case of plant cells, suppose it is a plant cell, mitochondria are present in both plant and animal cells. All the eukaryotes are having this mitochondria. But what about the plastids? Plastids are also double membrane bound organelles. So here we are writing the plastids, and these plastids are of different types. What are these plastids? Are different types. Leucoplasts. Anyhow, slowly we will study all these things. Simply in the names we are writing here. Leucoplasts are also type of plastids, and chromoplasts. These are also a type of plastids, and then chloroplasts. At least many of you know about this chloroplasts, right? So chloroplasts are also a type of plastids. So in general, I am writing plastids. So in this way, plastids are present here. So plastids are present in which organisms? Do all the eukaryotes have plastids? No. Plastids are present only in plants and euglenoids. Only these are having this. Only plants and euglenoids are having the plastids. But mitochondria are present in all the eukaryotes. This is what about the double membrane bound organelles. And then we are talking about single membrane bound organelles. Single membrane bound organelles. And what are these single membrane bound organelles? We will write the list here. So attached to this, there is a network like structure that is present, and this is called as endoplasmic reticulum. It is called as endoplasmic reticulum. Full name will be like this: endoplasmic reticulum. Anyhow, all these names already you studied in the previous years, means in the 8th class, 9th class, 10th class. At least you know the names. This is the endoplasmic reticulum. So endoplasmic reticulum is having only single membrane. This is completely a continuous one. It is a single membrane. That's it. So it is a single membrane bound organelle. Not only this, there are some other organelles called as Golgi complex are present. So in this way they are present, and this is called as Golgi complex. So this is Golgi complex, Golgi complex or Golgi complex. Anyhow you can pronounce this. Golgi complex or Golgi complex. This is the Golgi complex or Golgi bodies also you can say. They are also present. Not only this, they are having some small vesicle-like structure. They are called lysosomes. Lysosomes are present. These are also single membrane bound organelles. If you are talking about the plant cell, in the plant cell, suppose this is the cell membrane, it is having a large central vacuole. Is present. So that vacuole is also single membrane bound. And not only this, in the case of these eukaryotes, they are having some microbodies. What is the meaning of micro? Small organelles are present. So microbodies are present. Under these microbodies, what are important? So in NCERT, at least peroxisomes names was mentioned. So compulsorily you need to know about peroxisomes. These are also single membrane bound. Not only this, we will also study about glyoxysomes. These are also single membrane bound, and they are also type of microbodies. And also we will study about spirosomes. Understood or not? So these are some of the single membrane bound organelles. Next one are non-membrane bound organelles. That means there is no membrane for this. Non-membrane bound organelles. So these organelles do not have the membrane. Then what are those? Ribosomes. Some ribosomes are present freely here. So ribosomes are present here. They don't have any membrane around them. And there is one more that is called as centrosome. In each centrosome, there are two centrioles. So I am keeping the dots. What is the meaning of dot? There is no membrane there. So to say that there is something here, I have kept the dots here. So this one is called one centrosome. Please understand centrosome. Anyhow, when we study about the eukaryote, clearly we will study about each and everything. Completely it is called one centrosome. In one centrosome, how many centrioles are present? This is one and this is another one. So two centrioles are there. So one centrosome means two centrioles. Combinedly it is called one centrosome. Many students are making mistake here. They are thinking that centrosome and centriole are same. No. One centrosome has two centrioles. So these are the non-membrane bound organelles. And not only this, separately we will study about some other things. What are these things? Cytoskeleton we will study. Cytoskeleton we will study. And then we will also study about cilia and flagella. Cilia and flagella. Cilia and flagella. These are some of the things that we are studying with respect to the eukaryotes. And all these things we are studying under the heading of organelles. Please understand this. All these things we are studying under the heading of organelles. And here in the cytoplasm there are something else, but please remember this point was not mentioned in NCERT. Here also some inclusion bodies are present. Here also some inclusion bodies are present. Yes or no? So we human beings, so human beings means we are animals. We are also storing some food. We are storing the food in the form of glycogen, that is also stored in the form of granule, and that will come under inclusion bodies. But the problem here is in the NCERT regarding inclusion bodies, yes, they have talked for prokaryotes, but inclusion bodies in eukaryotes they have not mentioned anywhere. But still in higher classes you will study that even in eukaryotes also inclusion bodies are present. Okay, so this is the point. So anyhow, so this point since not mentioned in NCERT, so whenever it is needed then I will simply mention that these are the inclusion bodies but not mentioned in NCERT. Okay, so this is the overview for the eukaryotic cell. Please understand all these things. In the NCERT overview they have given even functions also. So what I will do is quickly I will tell you one function so that we can go for NCERT sentences reading. Anyhow, from the next lecture onwards we will go for regular explanation. So there we will cover all the points. Now let us see here. Cell wall, it will give the protection and it will also give shape to the cell. Over. Cell membrane, it is an open system, it is interacting with the surrounding, so it is taking so many materials into the cell and also it is releasing so many materials outside the cell also. This is the function of cell membrane. And then protoplasm, yes or no? This protoplasm is composed of cytoplasm and nucleus, and the nucleus is having two membranes and it is having nucleoplasm, nucleolus and chromosomes. Chromosomes are having genetic material DNA, and that genetic material DNA is controlling all the cellular activities. That's why nucleus is considered as brain of the cell. For the organism, brain is controlling organism. Same way, nucleus is controlling complete cell. So that's why nucleus is called as brain of the cell also. Okay, next one, cytoplasm. So fluid part is called as cytosol, and in NCERT for cytoplasm they have given two important sentences. Anyhow, it is a fluid matrix that already we studied. What are the two important points? Many reactions are occurring in the cytoplasm. So that's why they are saying that cytoplasm is the main arena, ARENA, arena. Cytoplasm is the main arena. What is the meaning of this arena? Arena means area, place. So cytoplasm is the main place for cellular activities in both plant and animal cells. This is the sentence they give. In both plant and animal cells, cytoplasm is the main place where many reactions are taking place. But here you need to notice one thing, not just for plant and animals, for all living organisms. For all living organisms, many reactions are occurring in cytoplasm. So that's why cytoplasm is the main area for all the living organisms, not just for plant and animals. For all living organisms, it is important. And next point, they are saying that many reactions are occurring in cytoplasm. Why they are occurring? To keep the cell in living state. To keep the cell in living state, that means cell has to survive. To survive, it has to do so many reactions. It need to produce energy also. So many of these things are taking place in cytoplasm. So many reactions are occurring in the cytoplasm to keep the cell in living condition. This is what they are talking. And next one, organelles. Different types of organelles are present. Directly we will go with the organelles. And what are the functions of these organelles? Here, double membrane bound cytoplasmic organelle. Please remember, these are all cytoplasmic organelle, but nucleus is not cytoplasmic organelle. Many students are making mistake. Nucleus is double membrane bound, okay, but nucleus is not cytoplasmic organelle, because now you just see under the heading of cytoplasm, will you write nucleus? No. Nucleus is protoplasmic organelle. Nucleus is cell organelle, but nucleus is not cytoplasmic organelle. But here whatever the organelles we are studying, all these organelles are cytoplasmic organelles, protoplasmic organelles and even cell organelles also. Understood or not? So in this way, nucleus is not cytoplasmic organelle, but it is cell organelle and protoplasmic organelle. It is part of this, but it is not part of cytoplasm. So that's why when you are reading the sentence, when you are making a sentence, when you are reading a question in examination, you should be very much careful. If you are thinking that all of them are same, again you may make mistake. So there are some small differences. Notice the differences so that you can understand easily. So nucleus is protoplasmic organelle, cell organelle. But these organelles are cytoplasmic, protoplasmic and even cell organelles also, because they are part of all this. So that is the reason. Understood or not? Now we will quickly go with what are the functions of these organelles. Mitochondria, mainly involving in aerobic respiration, so that ATP, energy production, will occur here. And leucoplasts, they will store food, mainly starches. And chromoplasts, they are having some color pigments, so that they are having different other functions that we will study separately. And chloroplast means they are helping in photosynthesis, they are, so that they are able to produce the starch and all. So that is the function. So this is the overall for the plastids over. Next, endoplasmic reticulum, it is synthesizing different types of compounds like proteins, lipids based on rough endoplasmic reticulum and smooth endoplasmic reticulum. And then Golgi complex, it will receive material from endoplasmic reticulum, and here some new changes, modifications will occur, so that now it may release the molecule to lysosomes, or it may release the molecules to vacuole, or it may release the molecules to plasma membrane, or it may release the molecules outside the cell also. In this way, Golgi complex is very important. Anyhow, all these things we will slowly study. Lysosomes. So as soon as a pathogen is entering into our body, or a food molecule is entering into our body, then some of the cells will engulf that particular pathogen or food molecule. And then what happens, once it is entering into the cell, inside the cell, lysosomes will go and bind, and then they will digest it. So in this way they are helping, and they are also having some other functions also that we will see. And vacuole, we are observing in the plant cell. In the plant cell, vacuole will help in osmoregulation. If excess water is entering into the cell, or some loss of water is taking place, now vacuole is very important in maintenance of this. So that is what the major function, and other functions also it is having. Peroxisomes, glycosomes, spirosomes, they are having different other functions. Separately we will study. Ribosomes, main function is protein synthesis. Centrosomes, mainly they are involving in cell division. In this way, different functions that they have mentioned. Understood or not? So this is what about the overview of the cell. And inclusion bodies not mentioned in NCERT, and you can call glycogen granules as inclusion bodies, that is a storage food in the case of animals, fungi and even eubacteria also. Okay, so in this way different things are the information that they have mentioned in NCERT. That too, in NCERT sentences they are specifically talking about ribosomes. So once we are completing about the ribosomes, then we will stop this overview and then we will quickly read NCERT sentences. And with respect to eukaryotes they are talking. In the case of eukaryotes, location of ribosomes they have given, but anyhow, when we are separately talking about eukaryotes, I will clearly give you the explanation. This is just to understand NCERT sentence I am giving this. Some ribosomes are present freely in cytoplasm, whereas some ribosomes, suppose this is the mitochondria, some ribosomes are present inside the mitochondria. This is the mitochondria, and this is the cytoplasm, and some ribosomes are present inside the plastids. Inside the plastids also they are present. And whereas some ribosomes, suppose this is the nuclear outer membrane, so this is the endoplasmic reticulum like this, this is endoplasmic reticulum. Some ribosomes are attached on endoplasmic reticulum also. So in this way, total 4 locations they are talking in NCERT. What are those? First one is present freely in cytoplasm is the first one, and present inside, in, this is in, but this is on. Please understand this. Present inside mitochondria. Third one is present within the plastids, inside the plastids. And the fourth one is present attached on endoplasmic reticulum. These are the 4 locations that they have mentioned in NCERT. Not only this, later on at the end of the chapter they are talking about nuclear outer membrane. This is the outer membrane of nucleus. Yes or no? And there also they have given in the nucleus part separately they gave Even the outer membrane of nucleus is also attached to ribosomes. This is another important point. Anyhow, under the ribosome separately I will tell you, but please remember this. Here also it is there. So actually 5 locations you need to remember for examination. This is what the location, this is the overview. So since it is overview, I don't want to give more details here. Separately we will study. If we study about all these prokaryote and eukaryote things, whatever we have discussed now, with that this chapter will be completed. Okay. So in this way, but this is a very big chapter and its weightage in NEET examination is very huge. Huge number of questions they are asking. So that is the reason why it is a very important one. And now let us quickly go with overview from the PPT. And then finally we will read NCERT sentence. Quickly we will go this. This is the overview. Different types of components are present in this cell. So here eukaryotic cell, if you see, cell envelope is having, see, glycocalyx is optional. Please understand, glycocalyx we are talking in prokaryote, but we are not talking in eukaryote. And then cell wall. In majority of the prokaryote, cell wall is there. Mycoplasma is not having cell wall. But in the case of eukaryotes, fungi, plantae and some protists are having cell wall, but remaining are not having cell wall. But plasma membrane compulsory for both prokaryotes and eukaryotes. Clear? And inside this, in eukaryotes we are using the term protoplasm. In prokaryotes we are using the term directly cytoplasm. And in the case of prokaryotes, nucleus is not present. But in the case of eukaryotes, yes, nucleus is present. Prokaryotes do not have nucleus, but eukaryotes have the nucleus. And if you are removing the nucleus, remaining content is called as cytoplasm or ground plasm. And within this again, only fluid part is called as cytosol or cell matrix. It is also called as hyaloplasm. And along with this, it is also having some organelles. In the case of prokaryotes in NCERT, they are even mentioning about inclusion bodies also. In this way, this is about the some general overview that we are studying. And in the NCERT, they have given a sentence here. For the plant cell, they have taken an example of onion peel. Onion peel is having, this is onion is nothing but a plant, right? So that from that peel you can isolate the cells and you can observe it. So in that case, from the onion peel cell you can observe outermost one as cell wall, because that is a plant cell. Suppose you are taking human cheek cell inside the buccal cavity, you just simply scrape it, you will get some epithelia. So if you are observing those cells, for those cells outermost boundary will be plasma membrane, because animal cells do not have cell wall. This is one point. Plant cell outermost layer will be cell wall, but for animal cell outermost layer will be cell membrane or plasma membrane. This was also mentioned in NCERT, so remember this. And here in the case of bacteria, outermost layer is the glycocalyx. In the prokaryotes we will study separately. For the same glycocalyx, if it is loose sheath, it is called slime layer. If it is thick and hard, then it is called as capsule. But anyhow, this point separately we will study under prokaryote. And cell wall, and it is the outer boundary for plant cells and even for bacteria also. Yes or no? This is what about this one. And here if you are observing a plant cell, if you are observing a animal cell here, for the animal cell, so this is the plant cell. For plant cell outermost boundary will be cell wall. For the animal cell outermost boundary will be plasma membrane, because here cell wall is not present. Okay, so for human cell also, because human cell is animal cell, so outer boundary will be plasma membrane. The next one, what is protoplast? So with equation also I told you, what is protoplast? Protoplasm plus plasma membrane. Protoplasm plus plasma membrane is the protoplast. Suppose here it is the cell wall they have mentioned. How you will prepare protoplast for the plant cell? By removing the cell wall. Simply remove the cell wall. So it will become protoplast. And next, protoplasm. It is surrounded by plasma membrane in the case of eukaryotic cells. So cytoplasm plus nucleus is protoplasm. This was the image that was taken from the internet. So you can easily understand this particular one. Next, cytoplasm. It is also called as ground plasm. It is having fluid matrix called the cytosol, and it is also having some organelles also. And this fluid matrix occupies majority of the volume of the cell. So this is what the content they are saying. Next one, these are the NCERT sentences. It is the main arena. Cytoplasm is the main arena. Arena means area, place. It is the main place of cellular activities in both plant and animal cells. Not just for both plant and animal cells, for all the cells it will be main place. Next one, various chemical reactions occur in the cytoplasm to keep the cell in living state. So different reactions are occurring in this cytoplasm. That's why cell is in living state, or else cell will die. So that's why to keep the cell in living state, many reactions are taking place in the cytoplasm. This is what the sentence meaning. And next one, so only fluid part is called cytosol or cell matrix or cytoplasm. And apart from this, some organelles are present. In the case of eukaryotes, membrane bound organelles are present, single membrane, double membrane, likewise they are present. But membrane bound organelles are absent in prokaryotes. This is what the sentence and its meaning. Next one, non-membrane bound organelles. What are those? Ribosomes. And these are present in all the cells, means both prokaryotes and eukaryotes, all the 5 kingdoms, all unicellular and multicellular organelles. These are common for all the living organisms. Next one. So non-membrane bound organelles which are helping in cell division of animal cell. What are these things? Centrosomes. Please understand centrosomes as a general sentence. If you say centrosomes are present in animal cells, absent in plant cells, but specific one separately in the regular content I will tell you. Okay. So next one. So these are the different types of membrane bound organelles. This is the endoplasmic reticulum. This is the Golgi complex, lysosomes, vacuoles, and this is the mitochondria, and these are all single membrane bound organelles. And mitochondria, chloroplast, these are double membrane bound organelles. And next one, cilia, flagella, these are seen in eukaryotic cells. And cytoskeleton, inside the cell they are present. Function, everything separately we will study. Cytoskeleton is also present inside the cell. And next one is regarding the nucleus. It is a dense membrane bound structure. Why it is a dense? Clearly you can see this under the cell. So it is a dense structure. So that's why it is a dense. And membrane bound. How many membranes are there? Double membrane bound. Two membranes are present. And this is present inside the cell. And next one, chromosomes are present in this particular nucleus, and inside these chromosomes genetic material is present. And what is the genetic material? Is it DNA or RNA? It is always DNA. All 5 kingdoms, all living organisms, all prokaryotes, eukaryotes, double standard DNA is the genetic material. Okay. Anyhow, slowly we will study about all these points. Next one, cells that are having membrane bound nucleus are called as eukaryotic cells, whereas the cells that are not having the nucleus itself, they are called as prokaryotic cells. So these are some of the important points you need to understand. Quickly we will read the NCERT content, and with that this lecture will be completed. You have earlier observed cells in an onion peel or human cheek cells under the microscope. Actually this was the experiment given in the NCERT for the 9th and 10th class students. So at that time, so they have already observed this. So that's why they are continuing this particular one. So the important point is onion peel, the outermost layer, the layer that you are taking out is called as the peel, right? So in that onion peel, onion is a plant cell, so it is having the plant cells. So for the onion peel cell, what is the outermost boundary? Cell wall will be the outermost boundary. Human cheek cells are animal cells. So what will be the outermost boundary for this one? Plasma membrane will be the outermost boundary. So this content you need to remember. Next one. Let us recollect their structure of this onion peel, plant cell, and a human cheek cell, that is animal cell. The onion peel, which is a typical plant cell. Onion peel cells are typical plant cells. They have distinct cell wall as its boundary. So what is the outermost boundary for the plant cell? Cell wall is the outermost boundary, and just within it is the cell membrane. Within it means inner to the cell wall is cell membrane, plasma membrane is present. And here the cells of human cheek have an outer membrane as a delimiting. What is the delimiting means? This is the outer limiting structure, that is the outer boundary. So what is that delimiting structure is called as plasma membrane. So in the case of animal cells, there is a membrane called plasma membrane that is acting as delimiting, that is the limit for the cell. So that is acting as the boundary. Next one, inside each cell is a dense membrane bound structure called a nucleus. Whether it is animal cell, plant cell, whatever it is, inside this a dense membrane. How many membranes? A double membrane bound structure, that is called as nucleus is present. And this nucleus contains chromosomes, which in turn contain genetic material, that is DNA. So inside this nucleus chromosomes are present, and within these chromosomes genetic material is present. What is the genetic material? For all the living organisms, the genetic material is DNA. But the term chromosome and the nucleus, these are applicable only for eukaryotes. They are not applicable for prokaryotes. In prokaryotes, nucleus is not there. DNA is present directly. There also it is double stranded DNA, but there it is directly present in cytoplasm. Please understand the differences. Cells that have membrane bound nuclei are called as eukaryotic, whereas cells that lack membrane bound nucleus are called as a prokaryote. So the cells having membrane bound nucleus are eukaryotes, eukaryotic cells, and the cells that are not having membrane bound nucleus are called as prokaryotic cells. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. Majority of the volume of the cell is occupied by semi-fluid matrix called cytoplasm in both prokaryotes and eukaryotes. That's what they are saying. The cytoplasm is the main arena, main area, main place of cellular activities in both plant and animal cells. Not just animal and plant cells, in all the living organisms, in all the cells, prokaryote, eukaryote, for all the cells, this is the main place where majority of the reactions are taking place. Next one, various chemical reactions occur in it. It means what? Cytoplasm. Occur in cytoplasm. It means, underline it and write cytoplasm. Occur in cytoplasm to keep the cell in living state. If these reactions are not taking place, cell will die. That's why to keep the cell in living condition, state means condition, to keep the cell in living condition, these reactions are taking place. In the next paragraph, besides the nucleus, what is the meaning of besides? Besides means along with the nucleus, the eukaryotic cells have other membrane bound distinct structures. Distinct, when you will use the word distinct, if you can clearly differentiate from others, if you can clearly observe that, if you can clearly see that, that is called as a distinct. What is the opposite word for distinct? Indistinct. Indistinct is opposite word, means indistinct means you cannot clearly observe, but distinct means we can clearly observe. These words are very, very important in the next lectures. So we will use these words. You need to understand these meanings. So there are membrane bound. When there is a membrane, then we can clearly observe them. So that's why these are membrane bound, distinct, clearly observable structures, and these are called organelles. Like some examples they are saying: endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbody, and vacuoles. And these are present in eukaryotic cells. Are they present in prokaryotic cells? No, they are not present in prokaryotic cells. The prokaryotic cells lack. LACK, lack means not contain, devoid, 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 lack. Both of them will give the same meaning. That means they do not contain. So prokaryotic cells do not contain such membrane bound organelles. So this type of membrane bound organelles are not present in prokaryotic cells. This is what the meaning is. Next one. Ribosomes are non-membrane bound organelles. What is non-membrane bound? No membrane. They are not having any membrane. Membrane is absent. Membrane is not present in this. So ribosomes are not having membrane. So that type of organelles are found in all the cells. What is the meaning of all the cells? Prokaryotes, eukaryotes, both. That means both eukaryotic as well as prokaryote. All the living organisms, all the living cells are having these ribosomes. Ribosomes are common for all of them. Next one, within the cell, ribosomes are found. Where they are found? Not only in cytoplasm. What is the meaning of not only? And then they are saying but also. That means these are the different locations, not just cytoplasm. It is not just only in cytoplasm. They are present in some other location also. Where they are present? First one is cytoplasm, they are saying. Next one, within. Within means inside. Inside what? Two organelles. What are those organelles? Chloroplast means plastids and also mitochondria. Inside the chloroplast, inside the mitochondria. And chloroplasts, are they present in all the eukaryotes? No, they are present in plants and also they are present in some euglenoids. That's it. Euglenoids actually they will come under protists. Anyhow, in the separate content we will study about this. So chloroplasts and mitochondria, they are having the ribosomes inside, in, within. Within word they have used. Underline the word within. And now they are saying separately and on. On means what? Above. Where? On rough endoplasmic reticulum. On rough endoplasmic reticulum. And next one, within mitochondria, within chloroplast, and then in cytoplasm. Not only this, we have added one more also. What is that one? Here itself we just write on, on outer membrane of nucleus. That also you need to add here. So in this way, total of 5 locations. On outer membrane of nucleus also you just add in NCERT. Okay, next one. Animal cells contain another non-membrane bound organelle. Please understand the content here. Animal cells, that means these are not present in plant cells. So another, why they are saying another? Already animal cells are having ribosomes. Ribosomes are non-membrane bound, right? Now along with this ribosomes, another non-membrane bound is there. What is that another non-membrane bound organelles? They are called as centrosomes. One centrosome has 2 centrioles, right? And what is the function of this? Which helps in cell division. This is the function of this particular organelle. So with this, we have completed this overview. So once at least you understand what are the different names and what are present in prokaryotes, what are present in eukaryotes, at least understand the names in this lecture. That is enough. From the next lecture onwards, we will study about prokaryotic cells in detail. Once we complete prokaryotic cells, then we will go with eukaryotic cells. Each and everything, whatever we have, at least the name we have used today, each and everything with complete details we will study in this next coming lectures. Okay, once we study all these things, this chapter will be completed. Okay, so this is how this chapter was given. So now we will go with next, in the next lecture we will go with prokaryotic cells. Okay, thank you my dear friends, we will meet in the next lecture. Thank you.